This is a quick summary of optogenetics. What is it and how does it work? Well, Chlamydomonas are a type of green algae that produces channelrhodopsins, and these let in positive ions that increase the EPSPs and thus increasing the chance for an action potential. They are stimulated by blue light and are tagged with the fluorescent protein, green fluorescent protein. Halobacteria. These are produced by Archaerhodopsins, and these remove positive ions from the cell via protein pumps that make the cell more negative. These act as neuroninhibitors, pioneered by Pastrana in 2011, and they are activated by orange or yellow light. Viral vectors. These are viruses that carry an opsin like channelrhodopsin, an identifier for a specific neuron type or promoter. These are injected into the brain or cell area of interest. And as you may know from the prefix opto, meaning light, lasers are used to activate ion channels that alter their activity. So shining a blue light in an area activates channelrhodopsins, and shining a yellow or green light activates Archaerhodopsins. Why use optogenetics? Well, it can stimulate neurons that can measure the effects of electrical stimulation on the cell. It can overstimulate the cell, which can kill it. It can target down to the neuron or circuit and can change the frequency of firing. It can stimulate in real time, is faster than neuropharmacology with great accuracy and precision. Yiza et al. In 2011 found that lasers can be used to work out the projection areas and brain circuitry. Optogenetics also has a bidirectional control, whereby neurons can be infected with both activator and inhibitor opsins. There are, however, limits to optogenetics. It can only measure as far as the light travels, and thus is only as useful as how far the light travels. Genetic engineering can't be done ethically with today's technology on live humans because it requires invasive deep brain stimulation. Perhaps in the future, it could be used as a depolarization block to stop tremors in Parkinson's. Optogenetics and anxiety. Fear causes an immediate subcortical response to threat. Visual or auditory stimuli reaches the thalamus and then the amygdala. The amygdala alerts the hypothalamus or HPA axis and locus coeruleus. This causes a sympathetic nervous system to release noradrenaline, decreasing ACH and the parasympathetic. As a result, a surge in cortisol and noradrenaline is produced, and it creates symptoms of increased blood pressure, tachycardia or fast heart rate, and sweaty palms. Subsequently, there is then cognitive processing of the immediate response to threat. So information is sent from the thalamus to the cortex and the basal ganglia for cognitive processing. The region then decides if the fear response should be continue or discontinue, if it's well warranted, that is. Roles of the amygdala. There is the basolateral and central nuclei, which is alerted in the amygdala, and fear responses are activated in the medial part of the central amygdala, the CEM, as well as the lateral part of the amygdala, the CEL. In the Nature journal, Tai et al. In 2011 injected a virus into the basolateral amygdala, where light was shone into the lateral part of the amygdala, which caused activation in the basolateral amygdala to the central medial pathways of the amygdala. It was used on rats in the plus maze and open area, which you can see on your right. Through this discovery, it was found that there were anxiolytic effects linked to the basolateral amygdala activating the lateral area of the amygdala, which reduced the medial output. Inhibiting the basolateral and lateral area of the amygdala increased anxiety. Thus, the basolateral amygdala activation caused the release of glutamate into the lateral area, and this stimulated GABA cells that inhibited fear in the medial part of the amygdala. Similarly, Knobloch et al. 2012 found that through linking channelrhodopsins to oxytocin promoter, this allowed for the targeting of oxytocin. They added the chemicals to the PVN, SON, and parts of the amygdala, and the researchers measured this through the freezing response in rats when they reacted to shock. It was found that activating channelrhodopsins in oxytocin terminals in the lateral part of the amygdala, or the central lateral area, reduced fear in rats. Oxytocin reduced shock, but this does wane over time, since shock does naturally produce fear. Optogenetics and neurobiology. Optogenetics can be used to understand the role of regions in drug abuse. For example, the prefrontal cortex and nucleus accumbens connection. The prelimbic cortex of the prefrontal cortex has glutamate connections that travel back to the VTA dopamine cells and nucleus accumbens. VTA standing for the ventrotegmental area. After adding cocaine intravenously, this blocks DAT or dopamine transporters and thus increases the amount of dopamine in the extracellular space, as in the synaptic cleft. After an extinction trial, a reinstatement trial is given and a laser is used to shut down this connection area. This then inhibits the prelimbic cortex, preventing drug-seeking behavior produced by re-exposure to the drug, to cocaine. Thus, we can tell that the prelimbic area or cortex is very important in drug-seeking behavior. So thus we can use optogenetics in the realm of neurobiology, neuroanatomy to map and track how neurochemistry reacts and leads to a change in behavior. Another example is Chen et al.'s 2013 Nature research. Basically, they wanted to assess the function of these prefrontal cortex neurons in the rats that displayed addiction phenotypes. Cocaine is resistant to inhibition by negative consequences. It is, however, hypoactive, in which inhibition of the pyramidal neurons drove the rats to take the drug despite being shocked. Activation reduced the cocaine seeking in shock-resistant rats and thus made them more shock-sensitive. Hence, reducing the activity in this pyramidal neuronal area of the prefrontal cortex, in fact, reduced the rats' ability to become aware of the shock, as they were relying more on the subcortical processing. While activating this same area, in fact, made them more aware of the shock, more sensitive of their environment, and thus inhibiting, or rather stopping them from taking or from singularly focusing on the drug. Finally, DREADs are designer receptors exclusively activated by designer drugs. These can target specific brain cells via viruses with the promoter. Muscarinic receptors, or HM3 or HM4 receptors, can be turned on or off using clozapine nitrous oxide, or CNO. Therefore, in conclusion, we reviewed what optogenetics is, how useful it is, its limitations, optogenetics and anxiety, what insights that we can discover, such as the inhibition of certain areas or activation of certain areas that help promote or reduce certain behaviors, since we are actually quite literally stopping neurons or activating neurons to work, and thus this can allow for more control, and looking at new ideas like the activation, exploring certain pathways, how certain drugs react to neurons in these certain pathways, and in other innovations like DREADs, designer receptors exclusively activated by designer drugs. I guess thanks for watching.